Hello, good morning and welcome to the Zoominar today. It's going to be the last one in the series. So it's a concluding Zoominar. We also see membrane proteins coming into this series by the speakers. Professor Gary Lorigan from University of Miami, Ohio. He always says that it's in Ohio as opposed to in Florida. And Kathleen Howard from Swarthmore College, Pennsylvania, Philadelphia. So before we get into the presentations, because today is the last one, I would like to ask Magda Ivanova to summarize the early career researchers' presentations and the award winners. Go ahead, Magda.
Hi, good day, everybody. It is my greatest pleasure to announce the winners of for the early career presentations. I just, because do you mind letting in the audience, in the panels, the winners once I start calling them? I just want to say that we are so glad to have the early career researchers from the early career to present in our Zoominar series. As you all know, they presented in 2 days in July and June of a total of 10 excellent talks. And they showed their great potential to do very challenging work and solve the challenging problems. And we were very excited to have them around. We had a really quite elaborate process for selecting the abstracts for presentations and for selecting the talks which were awarded. And in fact, we had 2 panels of judges. One was initially for selecting the abstracts, and after that selecting the talks. And the winners for the monetary prize are Emily Burt from the University of Leeds. I don't know if she's in the panel. Hi, Emily. It's just, I don't see you in a panel, but it's congratulations. It was really great to have you. The second.
Sorry for the interruption. She is there.
Hey, Emily, can you introduce yourself? And can you because let Frederica and Carmelo and Osashi in the audience after that? And Emily, can you say a few words about yourself?
Yeah, so I'm Emily. I'm studying my PhD at the University of Leeds with Professor Sheena Radford and Professor Frank Sobot. And my project is using structural mass spectrometry to study the aggregation process of alpha-synuclein.
Yeah, thank you. Thank you very much. And congratulations. Yeah, congrats.
For the opportunity.
And then Frederica and Carmelo, you can take turns. You had a joint presentation, so you can take turns to briefly introduce yourself, and you see you please unmute yourself.
Okay, can you hear me?
Yep.
Okay, I can start. So I'm Carmelo, and I'm a PhD student right now in Prague. I'm doing MD simulation and computational modeling on protein and membrane, and I'm working on human amyloids, amylin, and A-beta 140. And that's what I'm doing with Professor Carmelo La Rosa and Federica in Catania right now.
Yeah, thank you. And Federica, a few words.
Hello, can you hear me?
Yep.
Hello, I'm Federica Scollo, and I'm currently working in Prague with the group of Martin Hoff at Jaroslav Verosky of Physical Chemistry. And I'm working with the fluorescence technique and on amyloid, A-beta, IAPP, and also I'm collaborating with Professor La Rosa and with Carmelo as well.
Yeah, thank you. And Osashi, nice having you here.
Thank you so much. So myself, Ushashi Pramanik from IISCR Bhopal. I'm working under Professor Saptarshi Mukherjee. So my topic was the dynamics and folding pattern of an intrinsically disordered protein, which is bovine beta-casein, and the effect of folding inside a crowded environment. So it was really a great opportunity for me to be a part of this webinar throughout a year. Thanks to all the organizing committee and all the members, all the panelists. Thanks to all of you.
Yeah, thank you. And thank you very much, guys, for the exciting research and sharing it with us. And thank you to all the ECR presenters for really sharing their great findings. And I would like to thank the committee members and for doing such a great job in evaluating and deciding on the talks. And Rams, I'm passing it to you. Thank you
very
guys
much.
for being
Yeah.
here. Yeah.
Thank you very much, Magda and Bikash, for making this happen. Congratulations, you guys. You guys are the future. Great job. The prize money will come to you. I'll be in touch with you guys. We are waiting for one more sponsor. We have a sponsor lined up. So it's Journal of Physical Chemistry, and I'm waiting for one more. Thanks to the sponsors as well. So today being the last episode of this series, looking back, we started this quarantine, I call Quarantine from Reality series, virtual series in August last year in the middle of pandemic. It's now more than a year. It has been a wonderful experience listening to the speakers and high quality discussions from the participants. I'm sure that all of you enjoyed the Zoominars that provided a great deal of learning opportunities for everyone about a variety of exciting topics and latest findings, discoveries, and nice interactions with colleagues in the field. We tried our best to enhance the diversity among speakers and to involve early career researchers, including junior faculty, postdocs, and students. Obviously, this would not have been possible without the great speakers' contributions and their research team and their funding agencies and so on, and the energetic participants in this series, and the members of different committees that worked very hard in running the series successfully, and also the sponsor. So on behalf of the panel, I would like to thank all of you for all the great contributions to this successful series. And my special thanks to the panel, John Shea, Magda Ivanova, and Bikash for their great contributions. So by the way, all the videos of the presentations are available on a YouTube channel. Please feel free to use them. If you like, can cite them, you can use it for teaching and for other academic purposes. If you have any questions, you can contact me or other panel members. And if there are any mistakes that we made, I apologize on behalf of the panel. So today we have two great speakers, Professor Gary Lorigan and Kathleen Howard. First speaker is Professor Gary Lorigan from the Chemistry and Biochemistry Department at Miami University, Ohio. Gary completed his PhD in Chemistry and Biophysics from UC Davis and postdoc research at UPenn, where I met him. So I have been friends with Gary, right, Gary? He has been a professor of chemistry and biochemistry at Miami University right after postdoctoral tenure. Gary's research focuses on the structural studies of membrane-associated peptides and proteins using a combination of, I should say, unique combination of cutting-edge EPR, pulsed EPR techniques, and solid-state NMR spectroscopy. Gary has received many honors and awards. I will mention only two that I remember: the NSF Career Award and then Distinguished Scholar Award. And then he has served enormously to the community, the panels and funding agencies and so on, as well as the chair of their department, and now I think associate dean, or maybe dean, or maybe the president, I don't know.
No.
Not yet.
So
Okay.
please, enough's enough.
I'm reading the future then. Okay, Gary, with that, I would like to you to get started. You can share your screen and get started.
All right.
Do you see the share screen?
Yeah, hold on.
Yep.
Okay, well, thank you, Rams, for the kind introduction. Today, I'm going to kind of use a combination of using solid-state NMR and EPR studies to probe the structure and topology of the active and inactive forms of penholon. And my group, we use a combination of solid-state NMR and EPR. So here kind of shows you a picture. This is my 500 wide-bore solid-state NMR spectrometer over here on the left-hand side. And I think, as a lot of people know, that in order to study membrane proteins with NMR, we need to put a label on them. And usually we're going to go ahead and use an isotopic label like C13, N15, or deuterium, or we can use kind of natural abundance phosphorus to go ahead and probe the head groups and the lipids. So here's our 500 wide-bore solid-state NMR spectrometer. And then we do a lot of EPR in my research group. We use a combination of continuous wave, or CW EPR, in which, in this case, in order to study structural and dynamic properties, we need to attach a spin label. And what we'll do is we'll use kind of a standard MTSL spin label. We'll mutate out a certain residue, like an alanine to a cysteine, and we'll go ahead and attach that label to that protein that will report on that local environment there using CW EPR. And then one of the last things we've been doing for about the last decade is applying pulsed EPR techniques to study membrane proteins. And it's kind of a hybrid between the CW EPR and the solid-state NMR to study those systems. And down here at the bottom is one of my pulsed EPR spectrometers that we have here at Miami. So let's talk a little bit about penholon. So several years ago, I gave a talk at Texas A&M, and I met Rai Young. And Rai Young wanted to set up a collaboration with me to start investigating penholon, and he's one of the leading experts in the world on that. And so penholon is involved in cell lysis. It forms holes in the membrane upon oligomerization, and we're trying to investigate how this process occurs, trying to get structural and dynamic information on that system. Here shows you the amino acid sequence for penholon. There's two transmembrane domain ones. Two transmembrane domain. There's transmembrane domain 1, showed you here in blue. And then there's another one in green called transmembrane domain 2. Here shows you the R1 MTSL spin label that will typically attach and mutate out a specific residue. And how this process occurs is there's two transmembrane domains. What ends up happening is that transmembrane domain 1 translocates through the membrane, and what happens is it oligomerizes and it forms these holes in the membranes, and essentially a cell lysis occurs. And Rai's done some amazing work on this over the years. He has like the kinetics and the timing of it down, down to a science. He's mutated out numerous residues on this and compared different versions of that. And this is kind of the initial hypothesis of what actually had occurred. Transmembrane domain translocates and sits outside the membrane. Now there's another version of it. There's a couple different ones. This is anti-penholon, which kind of forms an inactive version in which transmembrane domain 1 does not translocate at the same rate at which the regular penholon version does. This particular version of the penholon, the anti-penholon, has a few different residues over here, the RYRS that's shown here in red. And in this one, it's much slower. It depends upon the kinetics, and in some cases, it just becomes kind of like an inactive version where it doesn't oligomerize as easily and doesn't form those holes in the membrane. So we wanted to kind of compare and contrast that. Now, my research group, we kind of have two ways at which we make our membrane proteins. We use a lot of standard molecular biology techniques in the lab, which we have other systems that are related to heart disease and potassium channel work. In this particular project, we go ahead and use a peptide synthesizer. So we have a CEM peptide synthesizer. We'll go ahead and make the peptides, and I showed you the amino acid sequence on the previous page. We then go ahead and perform HPLC to purify it. We then will go ahead and attach a spin label to it, and then we'll go ahead and put it into a membrane system. So since Kathleen Howard's here, I thought I'd just throw up a BICell picture because we did a lot of BICell work together as postdocs at Penn. So once we go ahead and put the protein into the membrane, we can go ahead and then do whatever biophysical techniques we wanted to do, whether we want to go ahead and do CD spectroscopy to probe the secondary structure of it, look at it with EPR with the spin label on it, or we can do pulse measurements like DEER, which we'll show you some examples here in just a few minutes. So here kind of shows you the first kind of study. We have two different versions here. We have the active version shown here in A that translocates through the membrane, and then kind of the inactive version in which transmembrane domain does not, or it's at a much slower pace. In a lot of cases, we like to go ahead and look at it from the membrane perspective using phosphorus and deuterium solid state NMR and see how the differences are between that protein. We'll kind of show you some of the results that we got from this. So we use some of these deuterated lipids to do those experiments. And here shows you kind of looking at the head group region where we're going to go ahead and do phosphorus solid state more on the active form of the protein. Where essentially what we're doing is we're looking at as a function of the addition of titrating in the peptide and also as a function of temperature. You can see that in each case, as we increase the temperature, the dynamics and the line shape changes, it becomes more mobile. In most cases, in this particular case, as we go ahead and titrate in the protein, which from 0 is the control with the absence of the protein, at 1 mole percent and then 2 mole percent, you'll start to see kind of an isotropic component that will grow in. So typically, this is what we'll see in a lot of cases. You'll see an axially symmetric phosphorus powder pattern, and you're going to see dynamic changes as we increase both the peptide concentration of the penholon as well as the temperature. Here kind of shows you know kind of a little bit of a difference between the active and the inactive forms. This is spectra shown at 35 degrees centigrade. The control sample is in black, and then the active shows you the 1 mole percent and the 2 mole percent. You can see that as we increase that protein peptide concentration, we start to see an isotropic component that grows in that's larger, and then you're seeing a shrinking in of that phosphorus chemical shift tensor, indicating that it's making some changes and interacting with the head group region of the protein in DMPC vesicles. The inactive version, you can see it's a little bit different. In red shows you the 1 mole percent and the 2 mole percent. You can see kind of a shrinking in of the phosphochemical shift anisotropy, but you don't see an increase in the isotropic component for the inactive version. Here shows you kind of the Deuterium Metamorph by taking D54 DMPC. In black, once again, is the control at 0%. You can see you get pretty well resolved quadrupolar splittings out of there. We're going ahead and we do depaking of it and calculate order parameters, but I'm not really going to show that today. We then go ahead and titrate in that peptide of 1 mole percent and 2 mole percent, and you can see dramatic changes both in the quadrupolar splitting and then the corresponding resolution that you would get when you do those depaking. And then here shows you kind of a similar as we did with the phosphorus NMR, comparing the penholon, the active and the inactive forms with 1 and 2 mole percent. You can see that the 1 mole percent, we do get some growth in that isotropic component in the same way that we had previously. You start to lose some of the resolution and the quadrupolar splitting shifts in. You don't see as much with the inactive version, the differences between the 1 and 2 mole percent, but it does kind of shrink in a little bit. And what we want to do next was kind of turn to Epr spectroscopy to look at it from the protein's perspective. So once again, what we're doing is we're going to put a label onto the protein. In this case, it's a spin label. And then we're going to go ahead and look at that local environment for each one of those residues. So for example, here, I'm showing you a series of CW Epr spectra of penholon, where we've gone ahead and taken a specific amino acid site like DR5 and replaced that with a spin label at position 1. And I've shown you there in green. And we're just going to do that for a whole series of the penholon. So 5, 8, 12. And what we're doing is we're kind of looking for changes in the line shape. And you can kind of see in certain cases, we're going to see 2 different components. And red would show you a more mobile, and then blue would show you a more rigid component. So we can go ahead and do that and probe all the different segments here of the penholon peptide. Here's another one that we can go through and see, going ahead and just comparing these. I'm not going to bore you with all the details on these, but the other thing that we can go ahead and do is kind of probe the structural topology of the peptide. Once we put the spin label into it, we'll do a set of experiments called CW Epr power saturation, where we can determine whether a certain residue with a spin label on it is located inside the membrane or outside the membrane. So what we'll do is we'll kind of do these experiments, 3 different flavors of it. In red, we'll go ahead and add nickel which is a paramagnetic species, which will help the sample relax back faster for the spin label and it'll give you a much quicker, a larger intensity that you'll see. So here what we're doing is we're looking at that central peak. So, oops. You can see here we're looking at kind of that central peak there from the hyperfine splitting that we see in the EPR spectrum. I skipped one over here. I'll have to go back. So in red, we'll go ahead and show you here with the nickel. You can see that this one is relaxing back and has much larger signal intensity. And what that means is that spin label is outside the membrane and it's relaxing back more efficiently with the corresponding T1 because it's outside and interacting with the nickel. If we compare that with the green, which is oxygen, oxygen is a paramagnetic species that's located inside the membrane and will help it relax back faster. And then blue is the nitrogen, which is kind of the control. So you can see in that particular case, the nitrogen and oxygen are fairly close in the power saturation, but the red is much larger. And that tells me that residue, the alanine 12, is located outside the membrane. And then we can look at another residue, which is 26, which is part of transmitter domain 1. You can see now the green is much larger. So in that particular case, that tells me that that residue is located inside the membrane. And then we have another one of transmitter domain 2 at position leucine 50, which is over here. And you can see also that the green gives you the largest signal intensity. That tells us that it's located inside the membrane as well. So this is kind of a way that we can map out the topology of the membrane protein. And let me go back. This one, the other thing that we can go do is probe the mobility of measuring the line width of that central peak. And we can kind of map that out for the Penhalon residue as a function of residue. So in red would show you the corresponding line width. That line width is directly proportional to the mobility of that spin label. The higher the number here in the mobility indicates that it has it's much more mobile, that spin label. So you can see the at the ends of transmitter domain 1 and on the other side of transmitter domain 2, it's very mobile, but then it's not nearly as mobile when it's actually embedded inside the membrane. So we can kind of map out the topology just by comparing those individual line widths. If we go back here and compare that for that central line width. And we can also do these power saturation experiments to help us actually probe which residues. And this helps us map out the topology of how the protein interacts with the membrane, which matches well with kind of what we see in comparison with solid state NMR. Once we go ahead and do a series of those power saturation techniques, we can then actually calculate a depth parameter for how far those residues are buried inside the membrane. In green shows you ones that are buried inside the membrane, and then red are ones that interact with that nickel that are located on the outside. And what you'll see here is in transmitter domain 2 and 1, we're seeing a slew of residues that are buried inside the membrane and red are on the outside. And here is in transmitter domain 1, you'll see kind of the mixture of red and green, indicating that portions are inside and portions are outside, which in a lot of cases indicates that it kind of sitting in the surface or partially embedded inside the membrane. Okay, so the other thing I'd mentioned previously for this Penhalon is that part of the process is that it oligomerizes. So here kind of shows you a, I need to go back one, it skipped one. Well, we're gonna use this DEER technique. So this is a pulsed EPR technique where we can measure a dipolar coupling between two different spin labels that are out to maybe, if they're in talking distance, from about 70 angstroms or so within each other. We can actually pick up the dipolar coupling between these two. We can use this if we put like two spin labels onto a protein, or if we have two different monomers that come together in close contact, we can actually pick up the dipolar coupling and probe the oligomerization state. And that's what we're kind of showing here with the Penhalon, where we're going through and mapping out some of these holes that you can see that form in this Penhalon. In this particular case, what we did is we went through and put a spin label onto our corresponding peptide at position 23. And we went through and did a titration of a protein versus lipid ratio. And green shows you like 1 to 1,000. Then we doubled the protein concentration and had 1 to 500. And then we further increased that concentration to 1 to 1 over 25. And what we're looking here is at our DEER signal. So the 1 to 1,000, we got very little deer signal from that, but the 1 to 500, you can start to see that signal intensity. This is this modulation parameter that we're looking at, the depth of this, of how far it goes down. And the red is at a much larger amount. So what this kind of lets us do is kind of compare our experiments and do them at different concentrations. So we notice at 1 to 1,000, we don't get very much oligomerization, dipolar coupling. So this allowed us to do a set of experiments where we had looked at it at a smaller concentration, okay? And then over here on the right kind of shows you some TE images that we went ahead and did of probing this pinhole as a function of this protein to lipid concentration. So here kind of shows you the deer results, where what we've gone ahead and done is put 2 spin labels onto, this is the active form of pinholin. And I think we did 5 different samples that we made. So for example, sample 1 would be residues 27 and 38. We collected our deer data over here, and this is the one in red. And we went ahead and got a distance distribution here of 24 angstroms for that. And 1 would represent, you can see there on the slide at the top, between residue 27 and 38. So 27 is on transmitter in domain 1, and residue 38 is on transmitter in domain 2. Sample 2, you can see, is a little further away, residues 24 and 40. So this is number 2. And once again, here we get a similar sort of distance, a little bit broader. It's about 25 angstroms. Then residue 3, we're looking at it further away. It's alanine 17 to alanine 38. And now we can see an increase in that distance of 35 angstroms. We then go through and do a couple more that's further away. And residues 4 is 20 and 44. And we get a distance of about 44 for that largest peak. And then residue 5, I'm sorry, sample 5 is residue 16 to 46. We get a larger distance. Okay. So then what we decided to go ahead and do was, let's repeat those same labels or close by, but then do it for the anti-pinhole, the one that's the inactive version that does not translocate through the membrane, or it does it at a much slower rate. So we went through and did deer experiments on that, and here shows you number 1, which is 27 and 38. We got a distance of 23 angstroms. 2, at 24 and 40, we got a distance of 25 angstroms. And then sample 3, we got a distance distribution there of 26 angstroms. We went through further and did this. We did 14 and 53, and we got 25 and 26 for 5, which is 8 and 53. So here you can see when we go ahead and span between transmitter domain 1 and 2, we're seeing very large differences in the corresponding deer data that we get. We can see in the anti-pinhole that all the distances are right around 25 angstroms as we go and probe. And if we go back here and look at our regular pinhole data, you can see that the distances, they go ahead and get larger as we scan the difference in that transmitter domain 1 and transmitter domain 2. And over here, I have kind of a summary here of the distances that we got here using this the deer technique. So deer is a really nice technique for probing different oligomerization states, different conformation states by using these spin labels. And so here in the active version, you can see we're getting huge differences between the different segments of transmitter domain 1 and transmitter domain 2. So 24, 25, and the distances get much larger, 35, 44, and then almost 50 there between 16 and 46. If you contrast that now with all the ones that we did for that inactive version of pinhole, you can see we roughly get about the same distance with an experimental error. So we're sampling both transmitter domain 1 and 2, and that indicates that there's very different conformational differences between the inactive and the active form. Both the inactive seem to be mostly located on buried inside for 1 and 2, excuse me. But then for the active version, as was hypothesized with Young, that it goes ahead and translocates through the membrane. And based upon our EPR studies here, that it looks like it's to be sitting on the surface. And that's kind of a distinct difference as to what Young had initially hypothesized, that sensory transmitter domain was completely outside the membrane. What we're seeing here when we go ahead and do use the NIH Explorer by putting in those different distances that we get and doing some modeling studies on this. Essentially, this is kind of what we get here for the active version in A, and then the corresponding inactive version in B. And this tends to match well with also our power saturation results for what we saw, as well as our NMR results previously and how they interact differently between the membrane, between phosphorus and deuterium and solid state NMR. Now, also, Rye's done a lot of experiments where he's gone ahead and compared essentially, he's done several different mutations of this S21 penholin. In a lot of cases, this is on the inactive, the anti-penholin version of this. And he's gone ahead and published some papers here on looking at different residues and seeing how it affects the corresponding timing and whether or not, for example, it actually will start to form the hole in the membranes and also the oligomerization of that. So we decided to go ahead and probe that a little bit using that DEER technique as well as some power saturation and EPR, CW EPR experiments. But here kind of shows you a pretty good summary of taking some of those residues where he saw differences. So in red shows you the one that we have here. This is a DEER technique, using the DEER technique where we put a label at position 8 for R1 and then at position 53 R1, and you can see that we get that 25 or so angstrom, difference. And then what we did is we looked at some of those other residues that he had gone ahead and studied here, and where he saw big differences, where actually in this particular case, you would actually start to see the formation of some of the holes and did these functional assays on them. So what ends up happening, like 17 is one of the key mutations that he pointed out. And what you'll see here in green is you'll start to see the shoulder kind of grow in over here, okay? And then we also did one for taking the same, so all of these have the same 2 spin labels at positions 8 and 53, and then we're mutating in some of those same ones that he did in his functional work. So the one in blue shows you what happens when you mutate out alanine 17 and then the glycine 21. And then the dark blue here shows you a further one that we went ahead and did. And what that indicates to us is we kind of have 2 different conformations that we're picking up. One that's kind of in the inactive, but then one that's actually forming more in that active conformation. And these mutations are causing differences in the translocation of that transmembrane domain 1 through the membrane. So we're working on more experiments on this, using a combination of NMR and EPR to go ahead and probe that using these powerful biophysical techniques. I wanted to give a short plug out for the Ohio Advanced EPR Lab that we have here at Miami University. If anybody is interested in collaborating with us, here shows you our 2 pulsed EPRs that we have. We have the one on the top is our X and Q band pulsed EPR system. The one on the bottom is our Q band only system. Both these are configured for DEER. We can do a wide variety of eSIM, indoor experiments, as well as a lot of different types of DEER. Our temperatures can range from 4K to 60K with both X and Q band. So I collaborate with a lot of people. Kathleen Howard, who is going to speak next, is one of our collaborators that we interact with as well here at the EPR facility at Miami University in Ohio. I wanted to go ahead and do the acknowledgements here. Indra, he's been a postdoc in my lab. He recently took a position at Campbellsville University in Kentucky. He still comes and uses Herrick. He's our EPR instrumentation specialist. Tanbir is a graduate student who's now a postdoc at University of Tennessee. Him and Dan Drew went ahead and did all the work on the penholin project that I talked about today. And my collaborators, Chuck Sanders, who goes ahead and I collaborate with on the potassium channel work that I didn't talk about today. But Rye Young is the one that goes ahead and did all the collaborative work on the penholin project. And then I need to thank the NIH and the NSF for financing the work. So thank you.
Thank you very much, Gary. Lovely talk. Great research. Let's give a minute or so for the questions to appear in the Q&A folder. I wanted to ask you that you showed initially the P31 deuterium NMR spectra of lipids, basically, with and without the pinhole in protein. I saw that even at 25 degrees Celsius, you see isotropic peak. What does that mean? Do you have free lipids, or do you have small vesicles?
It's just smaller vesicles. I think, that's one of those that varies sample to sample sometimes and how the sample's prepared. So the protein stuff we went ahead and did like numerous times, so we're pretty confident that I would say we did those in triplicate. So I'm pretty confident in those, but it can vary a little bit sample to sample depending on how you make your vesicles.
And then you also see a lot of, I might have missed this portion of your talk maybe. You see a lot of glycine in the TM domain, also serine and threonine. You talked about the S mutation, but threonine, so do you have any other role for this hydrophilic receptor, S and T?
Yeah, I mean, we've actually, done some, let me go back to show some of the different residues here at the beginning. It's got a lot of unique residues on it. Yeah, so here shows with a regular penholon. And we've actually tried to do some computational studies where you can go ahead and see the hydrophobic, like if you just change one residue. And we're actually doing that. When we first started the project, we would get conflicting results. So like a lot of cases when we make certain mutations in trimmer domain 1, we get different results. So initially it led to some conflicting information, but we're working on a paper right now where we're doing kind of what I showed at the end, but we're doing it with both EPR, CW EPR as well as DIR. So depending on what residues, some are more hydrophobic. You can see that transient domain 1 is one of those that's kind of tilting. It's kind of easy to go in the membrane or outside the membrane just by going ahead and changing, one or two residues. So.
What about the glycines? Do they have any role there? So many glycines are there.
Yeah, so if you look there at trimmer domain 1, there's definitely glycines that are in there, and yeah, they do play a role in there. Ryan goes ahead and talks about that in some of his papers. He's done several. I think he's mutated every single residue in there and done these functional assays on it. So
great. Are there any questions from the audience? Post your questions.
I didn't post one. Can I ask it in person?
Go ahead. Yeah, go ahead.
So I'm intrigued by DIR of homo-oligomers. So if you have a sequence and you label it and say it's a tetramer, you're not going to get a single distance. Obviously, you know this because we worked on it. You get the both diagonal and the proximal. So, and it gets even more complicated with higher order oligomers. I've seen some groups incorporate into their, when they're trying to process their raw data, put that information in there. So I think it's for a tetramer that the proportion, if it's a perfectly symmetric tetramer, it's you get one distance that's 1.2 longer than that. Have you done that at all? I mean, it makes the DIR look pretty ugly because it's not a single, despite the fact you put one label in there, you have a bunch of superimposed distances for say a single conformation. Have you guys worked on that at all?
Yeah, so there's kind of two ways to go ahead and do that is, so one where we went ahead and did the experiments that I showed you here, where we did it at a low protein to lipid ratio where we kind of minimize the oligomerization. I mean, you can see if I go back to, oops, oh, I can't, where's my?
Yeah, and so that's in a case where the oligomerization is concentration dependent, but lots of systems, no matter what the lipid protein ratio is, it's always going to be an oligomer.
So what we typically do in those cases is that we'll just do a cut experiment where we're going to mix it with unlabeled. So we would do it like a 1:5 ratio, and that way you minimize the oligomerization. We've done some experiments on that, but I didn't have time to show that data. For some reason, it's not. So this is what we did at the low protein. So that's what we did these experiments on initially. But we've done some cut experiments where we can go ahead and then probe, for example, what happens as you go ahead and explore those oligomerizations in more detail. But it does get confusing if you have an oligomer on it. There's no doubt about that. So
yeah, because even if you dilute it and you see a single distance, do you know it's the distance between its closest neighbor or across the oligomer? Yeah.
It depends on how big that hole is. So that's like these
Yeah.
modeling studies right here. You can see they're like 16 angstroms apart over there through that hole that it forms. It
Yeah.
just depends on, tetramer, heptamer, whatever it is. So.
Yeah. Thank you.
Sure.
So Gary, I have another question. Could you comment on the role of lipid properties? For example, if you vary lipids, you use just simple DMPC. What happens if you vary the lipids?
Yep.
What happens to the hydrophobic mismatch between the TM domain and the bilayer thickness, curvature? And could you comment on that just for the sake of completeness?
So the answer is we have seen some differences depending upon, like, if we use POPC versus DMPC. When we first started this, we were doing DMPC. We're doing like a whole nother set of power saturation with POPC, and we do see some subtle differences in that. So, and also the charge residues play a kind of a different, like if we had POPG in it in certain cases. So I don't have any of that data here, but we do see some differences, Roms.
So do the difference between DM and PO lipids, obviously thickness is different, huge difference there. Do you see that helix to be tilting differently?
We haven't gone in that sort of detail. In a lot of cases, we'll see like some subtle shifts, like in the deer spectra, for what we've done, but we haven't mapped it out careful enough to make firm conclusions about it.
So
and we have seen some differences, like in power saturation a little bit and line shape a little bit. So
good. Okay. There's a question from Walther. What do you think? What causes the completely different orientation of the pinhole-in and anti-pinhole-in as they only differ in the N-terminal region? And then
yeah, it's those. Let me go back to that one.
He says, I could understand that in nature, the TMD1 is hindered from flipping out, but by reconstituting the protein, it should not differ so much, as it is not inserted in the membrane in a defined membrane orientation.
Yeah, I'm trying to look for okay. So here, so Rai Young, he did a whole slew of experiments on this, where he went through and, he's the world expert on this. And the two different, so here shows you the Penholland amino acid sequence of TMD1, TMD2. The difference here are these residues, the RY, IRS. So when you add in this particular set of residues, it immediately slows down the transmembrane domain. It's because of the charges on here that go ahead and slow this down considerably. And in our eyes, when we went ahead and I should say, when we went ahead and make these peptides. So once again, these are peptides that we made, and we're putting them in, lipids that we buy from Avanti. What we're seeing is, essentially it not moving out. But these, the R residues here with these plus charges are slowing that down, or it not occurring at all. I mean, in his scale, it slows it down significantly. In ours, we're not really seeing much transmembrane domain location. It looks like they're both buried inside the membrane based upon our power saturation experiments. So it's this RYIRS sequence here that we add on to form that anti-penholland. But he's done a whole slew of experiments on this on the functional side. So.
Oh, great. Thank you very much. Thank you very much, Gary, for the lovely talk.
All right. Should I go ahead and introduce Kathleen?
Yeah, go ahead. Yeah.
Okay. So it is an honor to go ahead and I have two great friends here today, Roms and Kathleen Howard. And I met Kathleen Howard when I moved from California out to Philadelphia. She's one of the nicest people that I have ever met in my life. Definitely one of the nicest people I've met in science. Kathleen got her degree in chemistry from Princeton University. She then went ahead and got her PhD at Yale University. She then postdoc at the University of Pennsylvania in the Epr Lab, where Roms, myself, and her worked at in the late 90s. And after she did her postdoc at University of Pennsylvania in Stan's lab, she went through and became a professor at Swarthmore College there nearby in Philadelphia. So I am excited to introduce her, and I'm looking forward to her talk. So Kathleen.
Thank you, Gary. Let me share my screen. It's giving me an error. me see. Ah, there we go. Let me play around here a little bit. That is not,
Okay.
I'm having some issues with my stop share. Let me see. There we go. Come back. See if this works. How's that?
Yeah, we got it.
Excellent. So it's a great pleasure to be here today. Really thankful to ROMS for organizing this Zoominar series, and it's especially fun to be alongside Gary talking today. So today I'm going to tell you a little bit
Hey, Kitty,
of.
can you put it in presentation mode, please?
Oh, it isn't for me.
Oh, it's not showing it for me.
What's it look like to you?
You can just see, we can still see it, it's fine, but
No,
it's
I
just
want to
not
make it.
full.
Yeah, it is not in the presentation mode.
For me, it is. Let me try again. Let me try. Thank you for letting me know. Now let's see if it works. How's that?
Nope.
Really? It was working when I showed you guys before, right?
Yes.
Let me. Weird. You know what? Sorry, I'm going to just exit and come back in again. Sorry for the delay. Share. How's that? Does that work?
It's the same. It's the same, but we can see it. It's okay if you can't get it. So we can see it fine.
How about trying to
Sharing is
click the slide?
paused.
Can you click on the menu slideshow at the top in your PPT?
In PowerPoint, let me check.
Go up in the panel on your slide.
Mei Hong has suggested that if you're mirroring the laptop screen, then you might have to switch to the two screens.
So in PowerPoint, you're saying, what do I need to do?
She said, are you on an Apple Mac
Yep.
laptop?
Yep.
She said if you're mirroring the laptop screen, then you may have to switch to the two screens.
Is it in advanced? This is a bummer because I checked this before we did it. I'm going to do my desktop and then do this.
Try switching it there.
Is
Yeah.
that good?
Yes.
That works?
Yep.
Yep.
Excellent. I'm going to go back to my first slide then. Wow, this has never happened to me before. Okay,
Yep.
how's that?
You're good. You are good.
Okay, so I'm going to tell you a little bit about work we're doing on two influenza proteins that operate on the surface of the virus. So here's a cartoon of the virus in the upper right-hand shoulder. I'm going to put my mouse on here. And so why we're interested in these two viral proteins is they're essential in the infectivity process for influenza. And I'm going to start off with an inspirational little video, and this is just a clip. And before this clip occurs, a virus has infected a cell, probably a respiratory cell, and it goes into that host cell And then it hijacks the cell's own machinery, very tricky little thing, to reproduce and make lots of parts of the virus, where those parts then get transported to the surface of the infected cell and come together and go on to spread the virus. So I just want you to listen for the boom in this video because that's the specific spot of the infection cycle that we're interested in.
They get covered up and head to the surface where they get new keys, and then boom, here they come. This is an eruption of virus after virus after virus after only one virus entered the cell. But how many came out? Well, millions.
Yes. So we're interested what's going on. See these little new viruses coming out. What happens as they butt off the surface of these infected cells, and how does the genome get properly packaged inside these new viruses?
They get covered up.
Okay, so before I jump into the talk, I just wanted to give you an idea of how our research is done at Swarthmore. So it's an undergraduate only institution. And I've actually had the pleasure of working with a large number of phenomenal students. The students pictured here, there's 6 undergraduates, and they were the ones who worked in my lab this summer. You might wonder why I'm listing students back more than a dozen years, because I'm talking about 2 proteins today, and one of those, the very first student who worked on it was Krishna, and the work from these beginning students was super foundational to sort of building upon what we do today. And I also wanted to acknowledge up front, the beginning of this project on viral proteins began with a sabbatical I did in Bill DeGratto's lab. We've also had a number of really great collaborators that I'll talk about later on in the talk. Okay, so let's just talk a little bit more in detail about the flu life cycle. So it gives you an idea of why these 2 proteins we're working on are important. So as I mentioned on the first slide, the virus itself, this is a cross-section. It is a bilayer encased virus, so it's lipid bilayers, and it has a number of proteins stuck in it. And when it gets engulfed into a cell, there's a pH dependent process where the virus is induced to break open. It falls apart, and then the genetic material gets released into the cell. And as I said earlier, it hijacks the cell and goes here to the surface of the infected cell. And so it's a pretty fascinating process and surprisingly poorly understood for how exactly this assembly process works. And the 2 proteins I'm going to tell you about today, one is primarily involved in causing the curvature here, and the other one binds to that protein, and then it binds to the genome, and is important for pulling the right amount of the RNA into the new virus. Okay. So just another zoom in. So what are these 2 proteins? M2 protein, which is gets a lot of attention in the membrane protein literature. So it's in a relatively low copy number in the virus. It's the green little stalks here. But despite being in low copy number, it's completely essential for the viral cycle. What's kind of cool about M2 is it has multifunctions. Like a lot of viral proteins, viral genomes are minimal, so a lot of the players in there have multiple functions. So it acts as a proton channel early on and is key to that pH-induced breaking open of the virus. And what I'm going to talk about today is the interaction of M2 and M1. So M1 are these little lima bean shaped things that are lining the inside of the virus. It is actually the most abundant protein in a viral particle. And it also is the most highly conserved protein in the genome. And it's this interaction of M2 and M1 that we're pretty fascinated with. There's a lot of indirect evidence in the literature, biochemical, genetic and stuff that talks about this interaction, but nobody yet has been able to capture a complex of the 2 things and talk about that interaction at the atomic level. So why are we interested in this? Probably don't even need this slide, but there's annual flu epidemics, which we probably have all gotten the flu, but people have just learned to live with it. But particularly people who are immune compromised, it can be extremely dangerous. And as we're well aware at this point in history, viruses can cause pandemics. And early on in the COVID pandemic, people reflected back on in the COVID pandemic, about 100 years ago, there was a flu pandemic. And there was a lot of parallels drawn here. And interestingly enough, when people were predicting public health experts, is that it was thought that probably a flu-based, influenza-based virus would cause a pandemic over a coronavirus. But as we know, coronaviruses can do it. So clearly a huge public health issue. And then you think about, well, how do we combat viral infections? And we've thought about that a ton, the whole world. And clearly vaccination is super, super important. But as we've learned, when new strains come about, there is a lag before you can get a good vaccine. And even if you have a vaccine, as we know with the Delta variant for coronavirus, your vaccine's not going to work if you haven't adjusted it to the new strain. And even with seasonal flu shots that everybody gets, they have to make predictions the year before what strains will circulate. And often, despite their best efforts, they're only hitting about somewhere between 30 and 60% of the strains. So vaccination is super important. I'm big on flu shots. Yet you got to do other things. You got to wash your hands, do all those good things, clean well in the peak of flu season. Containment, we know a lot about wearing masks and social distancing. That's important. And I think what we've learned in this current COVID situation is that antiviral drugs are pretty important, okay? In that that lag before having a vaccine, and despite your best efforts at hygiene and containment, people are gonna get these viral infections. And it's been super difficult to get antivirals that work well. Viruses are very wily. They hijack the machinery of a host cell. And so you can't go after those things because then you're going to kill the cell. So viruses are tricky, but one thing that people have pointed at is that interaction of M1, which is highly conserved in M2. If we could somehow interfere with that process right there, that would be a really good antiviral target. Okay, so if we can clip it here, and perhaps you could make new viruses, but don't have the right genome in it because M1 wasn't properly bound to M2, or you can just stop that thing as well. That would be good. So long-term motivation for the work we do. So let me talk about M2 first. Our group's been working on that for more than a dozen years. So M2 protein, it's a 97 amino acid protein. Its functional state is a homotetramer. 2010, there was a lot of interest on this protein, and people went after it for a variety of reasons, mostly because at the time, it was known that M2 was a target for 2 antiviral drugs, and neither was effective anymore because all the circulating strains were resistant to it. But still, it was a druggable target and people were fascinated with it. 2010, here's a sampling of the structures that were published, and there was a perspective in Science that compared them and made some comments about membrane protein structures at the time. So the X-ray structures were done on chemically synthesized peptides. There was a solid state NMR structure that was done on bilayers sandwiched between glass plates. There was a solution NMR structure done in detergent micelles, and there was a paper that my group had published where we had done site-directed spin label EPR, as Gary told you all about, in liposomes. And over here on the top left, B here is a superposition of those. And there was a big discussion at the time, like, well, why don't they all look the same? And pretty clear why. We've learned more and more of the times is that, first of all, the different methods used give different information. And so when they are, the models are optimized, they're biased towards whether it was distances or membranes or things like that. The other thing was there's different constructs. And so the transmembrane had some differences from, here's another truncation that just went from the transmembrane up through like this first amphipathic helix up here. And then perhaps one thing that was pretty, became pretty evident at the time is those structures were very sensitive to the model membrane system. But right now I'm going to talk to you about work that our group did on EPR. So as we heard from Gary, the basic premise of our stuff is that we find a site that we're interested in on our membrane protein. We mutate it to a cysteine so we can covalently bond a spin label, the spin label, which Gary talked about as being labeled R1, gives a triplet here. So the spin half electron is split by the natural abundance N14, which is spin 1. So you get this triplet over here. I'm showing a picture of Krishna here. She was probably the first student we had that did EPR on the M2 protein. We were pretty excited. We got from NSF, we got our own CW X-band EPR, which is pretty unusual for undergraduate institution. And we've been able to upgrade it twice since then, but it's been a great workhorse for us. Krishna has since gone on to get her PhD at Hopkins, is now a senior scientist at Merck, and runs a drug development group. Okay, so I can go through this pretty quickly because Gary talked about it. So we put that spin label, which we refer to as a reporter group, on our protein, and we get a bunch of different information that's complementary. So you can get dynamics. If the line's pretty sharp, that means the spin label is moving around. It's probably out facing the water. If it's broadened, it's probably at a protein-protein interface or buried in the membrane. Really great information that's unique to spin label EPR, I think, is this idea of getting membrane topology. So as Gary talked about, oxygen is hydrophobic, partitions into the lipid part, as opposed to the aqueous environment. And spin labels that interact with that have specific relaxation properties. So in addition to oxygen, we use two other paramagnetic reagents. We work with a nickel complex that's in the water, so that gives us some information about the aqueous stuff. We've also gotten some great information. We use a lipid that has a covalently bound nickel to the head group, which gives us some very good resolution for what's going on the membrane surface. And finally, distances, super important for getting models for things. You can go from about 8 to 20 using just line shape differences in CW spectra, and then from 20 out to about 70 angstroms using pulsed EPR and Deer type methodologies, you can get longer distances. So what I chose to do today is I'm actually going to give you 5 little stories about what we figured out about M2 before I jump into the M1 interaction. And this slide speaks to a question that Ron's had for Gary, is this: we have figured out, like many others, is that M2 is super sensitive to the choice of lipid. So in this case, we very early step, we were using chemically synthesized peptides. I'm only showing 2 out of the 4 here just for simplicity. And we figured out you use lipids with short chain, so C12, as opposed to C18. The distance between spin labels within the oligomer changed markedly. Okay, so we could see that depending upon the lipids chosen for the bilayers in the model membrane system, you were going to get different structures, which was pretty exciting at the time because it helped explain why published structures were different. And if you just look back at different solid state structures that have been studied in lipid bilayers, depending on the lipid bilayer they chose, they were getting different helix tilts. Later on, we ended up using different M2 constructs. We started with the TM, and then we chemically synthesized longer pieces, put spin labels, checked them in different bilayer lipids, and found a similar thing about hydrophobic mismatch. We then went on and did a whole series of studies on lateral pressure profiles. So we used lipids with different head groups, whether big or small, and we figured out if we use lipids, PE lipids, we could work on like sort of making a girdle around the middle here, and we could change the conformation as well, which was pretty exciting. I wanted to put a picture here of Kei Seotomi. He went on and got his PhD at Columbia, and then did a kind of a superstar postdoc at Scripps, and he's actually been the first author in a series of papers that are crystal structures of membrane proteins, and then did some really phenomenal cryo-EM structures of membrane proteins as well. And I've gotten a chance to keep in touch with him over the years, and some of his earliest sample work that was key to getting his membrane protein structures went back to optimizing the lipid bilayers. So that was super fun to hear. So I mentioned earlier that M2 is a target for drugs. There's 2 of them that are on the market but rarely used because they have side effects, and most strains are resistant to them, but they look like plugs right here, carbon plugs here. There's a mantidine, and then there's a related one that's romantidine, when instead of having 2 hydrogens here, have methyls coming off of it. At the time we started working on this, there was a big controversy. There was a paper in Nature that showed 2 different binding sites, one within the channel, as well as some that were right on the surface and kind of at the little elbow that went between the TM and the amphipathic helix. So we jumped in because a bunch of those structures were done in biolayers that we thought weren't too physiologically relevant, and also we could get some more interesting information. So we made a whole bunch of different single site spin labeled studies. We studied dynamics, membrane topology, and a whole series of different things. We also worked on one of the peptides that was known to be drug resistant and pH insensitive. So that was kind of a neat control. So here's just one piece of data, which shows that if you look at spin interactions between sites, different sites along here in the TM, with and without drug, you can see a pretty dramatic change in spin interaction and thus the distances. So it turns out when you add the drug, the channel really clamps down and loses both dynamics and also more packing. I just wanted to throw in Jessica Thomaston. She left Swarthmore in my lab, and she went on to work for the DeGrotto lab, which is our long-term collaborator. She got her PhD at University of Pennsylvania, and then did a postdoc at UCSF. And she's published, I think now, up to 7 different crystal structures on M2, TM, and various different strains and drugs. So she had M2 in her blood and kept going with it. Okay, so I'm going to start transitioning to some of the ideas that led us to work on M1. So people knew that there was, studied a ton about the TM because that's where the drug was binding, but there was information that the M1 protein was binding to some place on the cytoplasmic tail of M2. And we were really interested in getting some information on that. The crystal structures don't ever really include past the TM for many reasons. And for one thing is that the distal part of this tail is pretty disordered under most conditions that people have studied it. But one of the things we did early on is to show in different bilayers, and particularly we optimize them, is that this y-axis is membrane accessibility. And so this is oxygen. So the more accessible you are to oxygen, the closest you are to the membrane surface. So this cool sine wave right here shows the amphipathic helix, where half of this side of the helix is membrane accessible, and then you go around to something that's non-accessible. And the periodicity of the sine wave is the periodicity of an alpha helix. We first started with chemically synthesized shorter peptides, and then we became molecular biologists, which was new to myself and the lab. And we've been overexpressing, purifying, and spin labeling for full length ever since then. Ani, which was actually the first person to do the topology work in my laboratory, is here. She got her PhD in chemical biology from Harvard, and works for a startup company in Cambridge right now. And here's Shen down here. He was the first one to publish spin label information on full length M2 in my lab. And now he is a venture capitalist who helps pharmaceutical companies get off the ground. Okay, so cholesterol has been a big issue in viral budding, because when the flu components go to the surface of an infected cell, they gather in these raft areas where the rafts are cholesterol rich. And there's have been a number of hypotheses that M2 has multiple conformations, and it shifts into a specific conformation that helps promote viral budding once it hits cholesterol rich membranes. And so we did a series of CW spectra in various different bilayers. And we noticed that they were multi-component spectra. Okay, so it's the superposition of two different spectra, and you can see I is for immobile and M is for mobile. We got really good at doing simulations of all different kinds. And so this is roughly what the line shape looks like for the mobile conformation. That's immobile, and you can add these in different percentages, and we were able to kind of rebuild and simulate the spectra. So the time scale of EPR experiments, if you can see those multi-component and CWs, it is two different orientations of the spin label, but it's hard to tell if it's just rotamers or if it's actually like a concerted conformational change. And we were able to answer that question working with Jim Fikes at the National EPR Center at Medical College of Wisconsin. This is saturation recovery data. So we made all the samples at Swarthmore, sent them out, and we worked with him for a long time. And basically, we could figure out from saturation recovery is that there was two different components in there. And the two different components were very sensitive to the amount of oxygen. Thus, those two different components represented two different conformations, which actually had different membrane topology. So that was cool. So we did a ton of information. This is just a little bit of it. Whole series of different sites. We looked at mobility, membrane topology. We did a bunch of deer. And we were able to show that, yes, M2 does have two conformations, two primary ones, and they are in conformational exchange. And they have different properties. Those arrows are dynamics, different properties of dynamics, different properties of membrane topology, and there's different distances in there. And once you put those, that protein into cholesterol-rich membranes, you actually get the same two conformations, but they're in different populations. So this conformation over here is much more pronounced than the other one. So that was pretty exciting. And this was our first taste at looking at conformational exchange using spin label EPR. The last little vignette on M2 I'm going to talk about was spearheaded by Alice Herneisen. And I have to mention her because she has been really important in our post-COVID recovery of our lab. So she graduated back in 2017. She's actually about halfway through her PhD at MIT. She's doing her PhD at MIT with the NSF grad fellowship. But when she left the lab, she had phenomenal notebooks and she wrote phenomenal protocols. So we lost the continuity between the upper level students and the younger students due to COVID, but we had her well-documented protocols, and she's on email and very helpful, so that was great. And also want to mention that her thesis project that she graduated with high honors from Swarthmore in is a cool question. So she was interested in the fact is that a bunch of people had done work on M2, but a particular budding defective mutant. And what happened is this M2 could lead to a little bit of curvature, but the buds never came off. The newly formed virus is stuck to the surface of the infected cell. And basically this mutant is they took 5 of the hydrophobic residues which were on the membrane facing surface, and they made them alanines. And in all the papers, it just said, oh, it's because this helix doesn't sit on the membrane anymore. And so they said it, and it makes sense, but nobody had actually tested it in vitro and was able to look at the structure. So she said she wanted to go after that and see if it was just as simple as that helix popping off the membrane. Turns out it was not that simple. There was a lot of things going on. I'm not going to show you all the data, but I wanted to throw in at least one deer spectra because we did this with Gary's lab. They were enormously helpful to us and worked on it. So we made a bunch of different, this is just a simulation of what a spin label looked like. So you just put a single spin label in there, but because it's a homotetramer, you get 4 spin labels in there, and you get, for a single conformation, you get 2 different distances, one between the neighbors and one between the diagonal ones. So it's 2 distances for a single conformation, you get 4 peaks for 2 different conformations. So it was kind of tricky. But just by looking at these spectra without even doing some analysis, you can see, wow, the difference between wild type and alanine is markedly different. So the entire catenary arrangement, the way these monomers are arranged within the oligomer, dramatically different. So we put a lot of information together, and we were able to come up with a model where that alanine mutant had a couple of things that were going on. So not only was that amphipathic helix jumping off the surface, that's what everybody predicted, but we were able to show that once that came off the surface, its conformation was completely insensitive to the presence of cholesterol, which was distinctly different from the wild type. And we know that sensitivity to cholesterol is important for targeting to the raft and also for a conformational change needed for binding. And we also noticed that without that amphipathic helix on the surface, the conformational change doesn't occur. So it ends up being the overall protein shape is more cylindrical rather than a wedge shape. And that wedge shape is well known in the literature for helping to cause membrane curvature. Okay. So we're ready to go on to M1, which is the focus of our most recent work. And as I said earlier, there's some interaction right here on the membrane surface. And the particular interaction that we're interested in is the one that occurs right before viral budding. So what do we know about M1? M1 is a 252 amino acid protein. Most of the information in the literature right now is for the N-terminal domain, which is 1 to 165. Pretty well understood. It's about 9 alpha helices packed together. What's a little confusing and makes the literature rather rich or complex is that the arrangement of these helices differ pretty much dramatically based upon pH, salt, and oligomerization state. So as you might have noticed from the previous slide, M1 forms ordered polymer around the inside of the virus. So, and depending upon solution conditions, it's found in dimers and tetramers and larger order oligomers. So that's something that we've been having to deal with all along. And the other thing was that based on the biochemical evidence is that it's thought that all the sites, I guess there's 4 of them that have been identified that are interesting for the M1, M2 interaction are in the N-terminal domain. So we have actually looked at both the N-terminal domain and the full length, but we're focusing mostly on the N-terminal domain. In terms of the full length, this is some Sachs model. And from sort of putting in the crystal structure, it looks like the head is about the same, and then the tail part here, and this is why it can't crystallize. It is pretty disordered, but has some different conformationals that it's different conformations it's samples depending on solution conformation. So first deal for us is that we actually had to get enough of this protein. We've been very lucky to develop a collaboration with the University of Potsdam, Salvo Chiantia. I was actually able to get there to Germany right before the COVID stuff happened and visit his lab. We've gotten 2 plasmids from him for different constructs for M1. And my students have learned all the joys of both expressing and purifying membrane proteins, which can be super tough, especially for beginners, but they have shown great fortitude. So this protein, because it has a propensity to oligomerize, it also aggregates, so it forms inclusion bodies and overexpression. And during sample handling, it periodically snows in your sample tube. But we've worked really hard to keep the protein at concentrations that are low enough so it doesn't precipitate, and also tweak the pH in the salt to do it. So we've been able to purify the proteins finally. And here's just a simple CD showing that it looks like the alpha helical protein that X-ray has shown us. So we've done a series of biophysical characterizations to show that we can form this complex of M1 and M2. And it's not trivial for anybody who's worked on membrane proteins and complex oligomerizing proteins. So here's a control. So we were following something that was similar in the literature. So what we did here is we made a mixture of different components. So the components we were dealing with was M1 protein, empty liposomes, and liposomes that we had carefully reconstituted M2 into. Okay, and so the first control was we just put M1 into the tube, we spun it super fast, and we looked at the pellet and then we looked at the supernatant. And the vast majority of that protein is still in the supernatant. We know that depending upon how we make these samples, some of the aggregates will precipitate out, but we're getting better and better at minimizing that pellet band. So the protein stays in the supernatant. Here's the M2 and liposomes. As we wanted, those pellet out, and none is stuck in the supernatant. So here's one of the controls, is that there's a series of different findings in the literature that even without M2 present, M1 has an affinity for lipid surfaces. And so we wanted to sort of figure that out, is that if we put the M1 into our M2 lipid systems, we weren't seeing non-specific interaction with the lipid surface. We were actually starting to see M1, M2 complexes. So, so the mixture just has the M1 protein. And so these liposomes don't have the M2. We see nothing in the pellet because the lipid doesn't show up on this SDS gel. And then the supernatant, the M1 stayed in here. So that was good for us. That meant that M1 wasn't specifically, significantly pulling down into the pellet. So this was the key one for us. So we made a sample that had both M1 and M2 in it. Okay, so M1, and then we looked at the pellet versus the supernatant. And so you can see here that some of the M1 is staying in the supernatant, and depending upon solvent sample conditions, we can get less of it in there. But we are getting some of this pull down into the pellet. So we are seeing an interaction of M1 with M2, and we're still optimizing this experiment. Okay, so how are we going to go about looking at M1 and M2? We actually have a whole bunch of different experiments, but what I'm going to tell you about today is the things we've done the most is that what we're going to do is we're going to look at M2 itself, which we know we can do, and then see if the spin labels, their mobilities, membrane topology, and distances change upon the addition of M1. So which sites to do? We have actually made over 35 single site spin labeled proteins. And so we had a choice of lots of things to look at. So we had to decide where we're going to put those reporter groups to look at the binding site. So the evidence in the literature is that something's going on at site 71 to 73, this red part here. So we picked reporter sites, 4 to start with, some that are before and 1 that's after. And we're going to go see if we can see any changes. But before, we had to tackle something that we had known in the lab was going to be an issue. So all our work on M2 so far, well, the vast majority on it is studied with our M2 protein reconstituted into liposomes, 200 nanometer liposomes. We actually published a paper where we went through a lot of very detailed ways about how to do that detergent mediated reconstitution to get reproducible, homogeneous, long-living samples. But despite the fact that the literature, some people who had done M2 had said it's unidirectional, other ones said, C-terminals in, other ones said C-terminals out, but we figured we need to check it for ourselves for our particular preparations. And lo and behold, it was bidirectional. So we did proteolysis. And if we were studying the interaction of the C-terminal domain and something we were going to add in, we knew that we were going to run into trouble because we'd always get spin labels on the C-terminal domain that were stuck inside the bilayer. We also knew from post-EPR work that we had done is that it's not optimal for these liposomes. Using smaller bilayer discs are useful. Also for CD, it's kind of a pain in the neck to do anything in 200 nanometer liposomes. They scatter terribly. So we had known for a while we wanted to work with bilayer discs. So as Gary had mentioned, I have a history in bicells. My PhD was about using bicells to study cell surface oligosaccharides. So we had done some bicells. We did another bilayer discs using an SMA polymer SMALPs. But for our particular samples of M2 in both bicells and SMALPs, we were seeing heterogeneous samples. So we've gotten pretty big on doing dynamic light scattering on all our samples before we do it to say, like, I want homogeneous, reliable samples. So we were pushing to do something, a different kind of disc. And so we have started making nano discs. So nano discs, this orange thing here is a membrane scaffold protein. And there's some great protocols in the literature for making those. And so advantages of our nano discs is that we clearly have access to both sides. And so we don't have any of the M2 binding sites that are hidden from the added M1, and also they're small. So we can do CD on them, they're better for post EPR. Okay, so current students, some of the biggest stuff we've been doing is making good nano discs. So nano discs in a nutshell, you start with your membrane protein and detergent. This comes right out of our purification protocol. You mix it with lipids, that's these green things here that have been also solubilized in a detergent. You mix these two with a membrane scaffold protein, and then you add in biobeads that pulls out all the detergent, and you get these assembled nano discs. There are a bunch of different parameters you can change. And so you have to be very careful. So we are doing all of our nano disc works in POPC, POPG. That is the same lipid composition we did all of our published M2 work on. Once we figured out which lipids we wanted to use, and we picked that because there's functional assays for M2. It actually acts as a proton channel when it's in that. There's a choice of different MSP proteins you can use, the membrane scaffold protein. We settled on one. It's actually the most commonly used one that makes about 10 nanometer diameter discs. You have to optimize the amount of lipid to MSP. We were able to find one that works well. And then you have to optimize the amount of your protein of interest with a membrane scaffold protein. We've done a ton of characterization. These three colors are the different proportions of MSP to M2. And then this is just size exclusion FPLC. When we look at the peak that comes off, then we do SDS on it and see we really don't want to be doing our EPR in empty nanodiscs or aggregated M2. So we were able to figure out which is the optimal proportion there. And then we've gotten, as I mentioned earlier, dynamic light scattering. And so we were able to settle on proportion of M2, and we say M2 is actually the homotetramer weight to 2 MSP proteins, which works out to 1 M2 channel per nanodisc, which we were pretty happy at. And now we can get beautifully homogeneous samples that last for a while. Okay, so 2 more slides. So what we wanted to establish now is that we wanted to show that our M2 conformation was just dependent upon its immediate lipid bilayer. Because we wanted to build upon the information we had done in liposomes already. And we had known from Bicell and Smalt's work before is that we were getting very different CW line shapes despite the lipids being the same. So here's just 2 examples of the spin label. One is in the liposome model membranes and one is in the nanodiscs, and you can see that they're superimposable. And then there's another site here. The signal to noise isn't as great in the liposomes, but they superimpose as well. So that was good news for us. And also we did the oxygen accessibility, so the topology is very similar as well. And so then the big experiment we're looking for is, could we detect, based upon this CW EPR, if we could see the interaction of M1 with our M2. And here's just one example. We've seen it on 3 or 4 different sites. But once you add M1 to these guys, you can see a dramatically different CW spectrum. And it looks like it's perhaps moving the conformational equilibrium to a different site. And then again, depending upon when you add the M1 there, you also have a different membrane topology. Okay, so what I'm going to show here in the last that previously I showed you ones that had all the different students, but these are the students that have worked on M1. And I particularly wanted to thank the 6 students who worked this summer. You know, it was a big deal to start up the lab after it had been completely shut down for a year. And of course, I want to thank Gary for the dear help he's done along the way. The people at MCW have been wonderful. Yeah, and Salvo at Potsdam. I'm really looking forward to continuing with you. And I wanted to thank all of you for your attention.
Great, thank you. We have time for some questions. You can feel free to post them in the chat. I had one question on that last set of experiments with the M1 and M2, what was I didn't catch what was the mole ratio that you used between M1 and M2?
So we actually did a couple. That one was, so nobody knows what the ratio of M1 to M2 is. Is it 1:1 per monomer or 1 to tetramer? That one was 1:1 M2 monomer to M1. Yeah, and so we're playing around with different stuff as well. So yeah, we're trying to figure that out with a couple different ways.
Yeah, the protein-protein interaction is always a lot more challenging, and there's a lot more you have to optimize. Have you tried with the spin label from M1 perspective too?
No, we would love to do that. In fact, we've started the design of that. So we were lucky with M2 is that somebody had actually already made a cysless version and had done some functional studies that I controlled that it still was a functional channel and some other things. But for M1, nobody has looked at that. So the M1 wild type has 3 cysteines in it, and it's not clear exactly how key they are. So we're doing that. We actually have started a system to do unnatural amino acids. So we will incorporate the spin label for an unnatural amino acid. So I have one student working on that as well. And in the whole theory of NMR and EPR stuff, we would love to put an isotope on one of them and look at the interaction with the spin label on the other. So yeah, that's the long-term plan. Rams.
It's a great talk, Kathleen. Nice work. Just going into your nano disc usage for EPR and reconstituting M1 or M2 protein. So what is the size of the nano disc? Is it like 10 nanometer diameter?
Yeah.
Or
so ours come out almost reproducibly. We can, if we calibrated both the size exclusion and the DLS, it comes out about 11.5 nanometers. With a lot more MSP in it, we see a big proportion of empty nanodiscs, but we're pretty convinced it's about 11.5 nanometers.
So do you see any measurement to show that the dynamics of the protein is retained, just like in vesicles?
Yeah, so that the line shapes, at least and the time scale that's relevant for CW, the CW line shapes are pretty much superimposable. I was kind of surprised because for bicells and smalls and stuff, they're really different. And that indicated to us, I wasn't quite sure why. Is it because there was perturbations between the edges and what was going on in the bilayer? But so far we've seen it basically, the protein just seems to know that it's in POPC, POPG. We haven't seen the difference yet. That doesn't mean we won't, but at least for the 4 sites we have, they look similar.
But don't you freeze your sample for measurement?
Excuse me?
Don't you freeze your sample for your EPR measurement?
No, so we've been doing everything at room temperature so far.
All right, okay.
Yeah, so we haven't done any here on that. So Gary, we might be sending your stuff your way. But right now, as you know, when it's frozen, it does look different. But that's another advantage is of the CW and stuff we can do. We do everything at room temperature in both the nano disc and that. And so that's where we do at the X band, we can do mobility, membrane accessibility through different relaxation agents, and some CW distance stuff.
Would you expect any potential influence of the belt of the nanodisk on your protein orientation? And
yeah, that's the control that we're trying to do. And that's why we're looking at comparing very carefully the membrane topology and the line shapes in the 2 of them. So what I could pull from my students, they all left at the end of July. They kicked them out for COVID stuff. I got 3 of them. I've seen all 4 of them that show that they're relatively superimposable. So according to their eyes, they're like, oh, they're the same. But of course, you know, I gotta check. But at least the one they sent me and the ones I've seen is that they look very similar in a way that hasn't jumped out before. So we're hoping that we can pretty reliably use what we learned about in liposomes to apply to what we've learned in nanodiscs, which are really much better for the M1, M2 interaction.
Great. Gary, you can open up the session for questions for both the speakers, including you.
All right. The floor is open for questions.
You can join the panel by raising the hand function. Raise hand function. Any questions from the panel? We got bigger panel today. Magda, go ahead.
Yeah, hi, very exciting question. I'm kind of looking more into the future, trying to think. I know that you're working with the lipid composition and stuff like that, but I'm not much of a virologist and stuff, but it looks to me the lipid composition, especially for release of the viral particles, is fairly important. I don't know if I'm correct. Can you comment a little bit on that? Just what is the next composition you will try to go after, and whether that is promoting or inhibitory of the viral production?
Yeah, that's an awesome question and things that lots of people are thinking about. And here there's the tension between in vitro homogeneous samples and physiologically relevant. So POPC, POPG with some cholesterol has been sort of our, what we stick with, because we know it's, we can reconstitute into it and do it well. But group like Mei Hong's group, they've used a viral mimetic membrane. So you can use other membranes with many more components that are indicative of the lipid composition of the rafts. So basically all the membrane, the flu proteins aggregate on the surface. They have to find each other so they can self-assemble. It's really phenomenal that they can do this. So they have to find each other, and they come together on a raft, which has a different lipid composition than the rest of the cell. So yes, you can do that. It's just harder, and it's harder to interpret stuff. So we've actually used the viral mimetic membrane, and to us, at least just looking at we have 1 or 2 papers when we did a whole bunch of different lipids. It looks like high concentrations of cholesterol, at least to us. So yeah, I always think of M2 a little bit like a transformer. I don't know if you guys had kids where you have like a little toy where it looks like a robot, and then you just change a couple of the angles and it becomes a truck. And you can rotate these around. So M2 can be in different conformations. And then once it hits a lipid composition for something where it's okay, now I'm a fission facilitator. Early on in the cell, it's a different lipid composition when it first goes into the infected cell, and it actually acts as a proton channel. You can say, oh, I'm going to flip into my channel conformation. So I like to think of it as having, clicking around. But I think you're absolutely right, is that the lipid composition right before it buds is completely key, and probably we need to do the best we can if we want to study M2, M1 to mimic what's going on. Not to make things even more complicated, but all viruses are, is that in a real system, you also have hemagglutinin and neuraminidase there. And both of those are shown to bind M1 as well. So gotta go into a little in vivo EPR. I think that's beyond the capability of my own lab, but yeah, we do the best we can.
Can I ask the kind of controversial question maybe?
Sure.
Where are we in the field in terms of the results from crystallography and the micelles, detergent micelle solution MR data, and then now we are, you guys looking in lipid bilayers, including Mei Hong. So where are we now? Which one is, how do you take this results from detergent micelles and crystallography? Are they agreeing with you guys, or how much different are they?
So my take on this is that a multi-pronged approach is the best one. So each one of these different methods contributes something different. And you just have to, it's okay if they don't give what you consider like a single model. We're all looking at it from different perspectives. And so I do believe the immediate environment, a bilayer is definitely part of it, particularly for a small protein like M1. So it is, it's known to have a lot of flexibility, so it really matters what the lipids are around here. So there's been some different information in my cells that you can't really, doesn't seem to agree with some of the bilayer stuff. That being said, there's some biophysical methods that only work well in those systems. So you take what you can get from that. And I think in the end is that instead of being, controversies, that we each look at the different perspectives you have and then treat it with, a certain amount of not disagreeing with it, but realizing that there's limitations for each one of these methods, for each one of these sample compositions, and then look at it holistically.
Yeah, thank you. So looks like Mei Hong, you can join the panel.
you want me to read it or?
Yeah, this is for Kathleen. Go ahead, read it.
Oh, I can read it. I see it. Or do you want to, you can read it, Gary, or you want me to read it out loud?
I'll read it out loud, then you can answer.
Okay.
All right, we can have Mei Hong actually ask it live in person. How's that? Save everybody the problem.
Hi, Kathleen.
Hi, good to see you. Thanks for coming.
Yeah, so two questions. One is, how does the M1 interaction with M2 impact the proton channel function in your mind? I mean, this is, just at this point, do you have any working model?
Yeah, so taking a big step back and simplifying things. So we're assuming we're most interested in what's going on at right before budding. And so we're assuming that it's not acting as a proton channel at that point. So it's high pH. And so we're probably totally simplifying over things, is that we're interested in the interaction right at that point. So we're only doing whatever we refer to as our high pH, pH 7.8. So we're, yeah. And the other thing is that we have some issues. M1's oligomerization state is very sensitive to pH. So we haven't been messing around with low pH with M1.
Okay.
Yeah, so I'm totally separating them in my mind.
Yeah.
So, and I don't know what the M1, there is some interaction, clearly when the virus falls apart in those early endosomes, there's probably something going on there. But we're trying to focus more on the last part right before fission.
I see. Then my second question is, you had a wedge model versus cylindrical model for the wild type versus the mutant. What experiments did you do to distinguish these two models? Seems like you would have to have a lot of distance measurements at different parts of the protein. Is that what you did, or?
So we worked a bit with DeGrotto and that, and I'm blanking on his name. Anyway, they did a group of SACS data. I don't know if you remember, so they did a whole bunch of different lipids and a whole bunch of different.
Yeah.
Oh, Nathan Schmidt. Nathan Schmidt.
Yeah.
And so a lot of that, actually, that's hypothetical. So we say no, there isn't a lot of distances there. That is a model, I have to say. And so there is some information there, is that we extrapolated from, there was some information on there, but we piggybacked a lot on their scattering work in various different lipid compositions to talk about the fact is that they made a hypothesis then, is that the reason why they got more curvature for full length as opposed to the transmembrane is that there was this interdomain coupling, and part of the C-terminal domain was leading to it to be more of a wedge. Yeah, so.
I'm more thinking of the non-wedge shape. When you mutate
the
traffic helix, and you think that the shape has become less curvature inducing, less a wedge.
Yeah.
That I'm curious about.
Yeah. So one thing that we did have there was distances for that. And so what was going on there is that we were looking at a site, L43, right at the end of the stop, and there is a dramatically different distance.
Okay. Which direction? Longer or shorter?
Shorter, which we were surprised at. We figured the amphipathic helix pops out, the whole thing would kind of flag out. But it ends up being that anchoring it to the surface changes the conformation of the transmembrane part itself. So that was really surprising to us, because I kept going back to Alice, collect that again, because it was backwards. We had thought amphipathic helix pops off, the entire thing gets more dynamic. But it wasn't true for the end of the transmembrane helix.
I see. Okay, so that's interesting.
So that was pretty interesting to us. And so that's why we got this idea of interdomain coupling with the N-terminal domain. What it's doing and its sensitivity to cholesterol and stuff does affect the transmembrane part. You can't just say the transmembrane does whatever it wants and ignores the C-terminal domain. So yeah, that was kind of cool.
Good.
It looks like we are through with the questions. Maybe time to wind up. Magda, do you have anything to say? If not, thank you very much, Gary and Kathleen, for lovely talks and great discussions. Thanks, May, for joining us, and thanks, UCR awardees. Congratulations, and be in touch with me, or I'll be in touch with you once I get the dollars, and you'll be very rich. I promise.
Yeah, thank you so much for everybody and the audience and the speakers, and it has been really great experience. Thank you for, we had lots of volunteer help here to make that run, right, Rams? It was
Yep,
really great.
yep.
And thank you for including me. I was really excited to be part of it.
This is the last one, or I didn't hear.
This is a concluding, yes,
I see.
last one.
Okay, I see.
Thanks to Kathleen
Yeah.
for having touchdown.
My pleasure.
Yeah, it was nice meeting you guys. It's really nice meeting you.
So thanks all very much, all the participants and registered participant speakers and sponsors and everyone on the panel, Vikash for the lovely videos and lots of hard work on that. With that, bye-bye. See you. Ta-da.
Thank you.
Bye. Bye.